In this video, we will discuss about the impact of Dasatinib in the survival probability and the metastasis and proliferations in the mice model or in the in vivo studies when the mice is infected with this PDAC, that is pancreatic cancer cells. So before directly moving into the studies, they also presented the results to uh, get the optimum concentration of dasatinib that can be used for the in vivo studies. So on the right hand side of the figure in the panel A, uh, they have shown the results for the different concentration of dasatinib, and they are able to see that at around 10 milligram per kg, they can see a reduced activation of this SRCC compared to the case where 5 milligram per kg dasatinib is used. And these things they have also validated using the immunostaining, where they can see that in the vehicle, the signals for the activated SRCC or the phosphorylation of SRCC at 418 is high compared to the case of dasatinib, where the activations of the SRCC is low. So from these experiments, they are able to uh, find out the optimum concentration of dasatinib, that is 10 milligram per kg, that will be used for the in vivo studies. So when they did the in vivo studies, in terms of the survival rate, what they are able to find out is that there is not much difference compared to the vehicle and the dasatinib that are injected in the mice models for different time intervals. That can be seen here, but they decided to check whether it has any impact on the metastasis and proliferations of the tumors or not, or the dasatinib has any impact on the metastasis or proliferations of the tumors within the mice models or not. So they check the in panel B, they shows the results for the metastasis, and they are able to see that the mice models that are infected with dasatinib for 6 weeks and 10 weeks has reduced metastasis compared to the vehicle case. And for in the paper they have shown that for the dasatinib that are infected for the 6 weeks has a reduced has a metastasis for like 26% of the case, whereas when the dasatinib is infected for the 10 weeks, they have a metastasis in 23% of the cases. And these things they have validated the reason for the metastasis using the Western blot, that the activation phosphorylations or activated SRCC is low or reduced in the case where dasatinib is used compared to the vehicle case. So that could be the reason why there is a reduced metastasis in those mice models. Thereafter, they also wanted to check whether it also has a role towards these proliferations or not. Dasatinib cause reduced proliferations or not. So for this, they use this BRDU, which usually binds to the newly synthesized DNA and can be find out using the S phase analysis using the flow cytometer. And what they are able to find out is that in the case of this BRDU, they can see a reductions in the S phase, reduction in the S phase compared to the vehicle case, and this shows that there is a reduced proliferation also in the case where dasatinib is used in the mice models. And these things in the panel E, they shows the result for this PanIN or the early pancreatic cancer stage, and for this panel F, they have shown the results for the this late stage pancreatic cancer. And in that case, they do not find out. In that case, they can see that the proliferation rates are higher, and these things they have validated using the immunostaining also, where they have shown that in the compared to the vehicle case, the proliferation rate in the dasatinib is lower in both the cases for these PANC-I1 cases, that is early stage of the pancreatic cancer, and the PDAC case, that is the late stage of the pancreatic cancer. So from this, they are able to conclude that although dasatinib has a role towards suppression of the metastasis and proliferations, but it does not cause too much impact on the survival probability of the mice that are used in the in vivo studies. So overall, this dasatinib does not seems to be too much helpful from the results that are presented in this paper. So if you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.